So thank you all for being here. My name is Alessia, and today we'll talk about lipidomics of lipid droplets in health and disease. I'll give a short introduction, we'll go through some processes, and then we'll see some applications. So in the introduction, we'll talk about what lipid droplets are. Then with the processes, we'll see how lipid structural flexibility affects their incorporation into lipid droplets. And finally, we see some case studies. So about the role of lipid droplets in starvation, in kidney disease and diabetes, in remyelination, and in ischemic stroke. We'll start with the introduction. So we'll see what a lipid droplet is, what is this normal structure and physiology. A lipid droplet is a highly dynamic organelle that is essential for the regulation of lipid metabolism, storage, and transport. So it's a specialized cell compartment that stores lipids and especially is useful for buffering toxic lipids and to prevent, for example, peroxidation. So the peroxidized lipids can be stored. It consists, as you can see from this figure, of a phospholipid monolayer, which has some specialized proteins embedded in it. And then there's a core of triacylglycerols and steryl esters. How does the biogenesis of lipid droplets start? It starts in the membrane of the endoplasmic reticulum, especially when there's a nutrient surplus. So you'll see that fatty acids and sterols will be stored into the lipid droplets as triglycerides, as we said, and steryl esters. So they start accumulating and in the space between the membrane, and then we'll start forming this lipid lens that will push and eventually bud out into the cytosol. The lipid droplets will start growing, and for example, smaller lipid droplets can be embedded into bigger lipid droplets, and finally, it will reach a steady state. And then when we need lipid remobilization, so when we need to, for example, glycerol or fatty acids for energy, or we need some specific lipids to generate phospholipids from membrane, this will be remobilized. And now we'll see how lipid structure flexibility affects their incorporation into lipid droplets. And this study was done by the Korea Research Institute of Chemical Technology. The research question, as I said, is how lipid flexibility affects its incorporation into the lipid droplet. And the researchers in this case were particularly interested in this medicinal lipids and this non-saponifiable lipids. So we see squalene and zeaxanthin. You can see from the figure here the difference between these two. So the squalene has isolated pi bonds, which makes its shape more like a worm, so more flexible. While zeaxanthin is more rigid as it has conjugated pi bonds, so it looks more like a rod. So they did some molecular dynamic simulations and did some microbial lipid engineering to see how these two structures affect incorporation into the lipid droplet. So first, as we said, we start with some molecular dynamic simulation, and you can see that the flexible lipids, so squalene, can enter the phospholipid monolayer and go into the tightly packed triacylglyceride core of the lipid droplet. While zeaxanthin, being more like a rod, becomes trapped into this phospholipid monolayer and cannot really penetrate into the core of the lipid droplet. So the next step was going to in vitro studies. So they used yeast cells that were genetically modified. They were producing zeaxanthin or squalene, and they were also engineered to have either overexpressed or deleted some of these 10 genes that were encoding for an increased lipid droplet size or increased lipid droplet number, as you can see. So they could see from the yeast cells that there was an increase in squalene content in the cells that were producing that. And this increased with the number of lipid droplets and also more with the size of the lipid droplets. And this is probably because the incorporation of squalene was making the triacylglyceride core of the lipid droplet like more leaky, so even more squalene could penetrate into the lipid droplet. Well, for zeaxanthin, you can see that there was an increase when the lipid droplet number was increased more specifically. And this is probably because there was a higher surface area in the lipid droplet that could allow for more zeaxanthin to become, as we saw, kind of trapped into the membrane of the lipid droplet. So we saw from this study that lipids with isolated pi bonds are more flexible, while lipids with conjugated pi bonds were more rigid. And this affected how they could migrate into the lipid droplet, where the flexible lipids could migrate more easily into the lipid droplet core. And so this is fundamental to understand how this could affect the lipid droplet formation and what actually can penetrate the lipid droplet. This study was published in 2022 in Nature Communications and was titled Chain Flexibility of Medicinal Lipids Determines Their Selective Partitioning into Lipid Droplets. So now we'll start with some case studies about the specific role of lipid droplets in different applications. And the first one will be about starvation and lipid metabolism. This study was done by the Center for Genomic Regulation, which is an international biomedical research institute comprised of different institutes from all over the world. In this case, the authors were from Spanish and American research institutes, and they were studying what the role of lipid droplet is in starvation and lipid metabolism. So we know that TANGO2 is a crucial protein, and it's encoded by the TANGO2 gene in humans, and as a role, for example, in cell activities like endoplasmic reticulum to Golgi transport, and also for the health of the mitochondria, to maintaining a healthy mitochondria. But there's no specific details yet known about its function. We just know when there's mutation in the TANGO2 genes, we see disruption of a lot of processes. We have like early symptoms like lactic acidosis, low blood sugar, high ammonia levels, and this translates into worse outcomes like microcephaly, cardiac arrest, etc., as we can see here. And we know that these outcomes get worse when there's also starvation conditions. So the first thing that the authors wanted to understand is where TANGO2 is located, and they saw it was in the mitochondrial membrane near the endoplasmic reticulum and also near lipid droplets. And they could also see that in TANGO2 depleted cells, the size of the lipid droplets was increased. So they thought there should be something different in the lipid metabolism of TANGO2 depleted cells. So in this case, as you can see here, we have a full lipid profile. This was done with our untargeted shotgun approach. This uses direct infusion mass spectrometry, and we can see a lot of lipid classes in one single analysis, as you can see from this graph. And in this case, for example, you see the wild type cells under starvation condition on the left side of the graph, and a lowering of lysophosphatidic acid and an increase of phosphatidic acid. While the opposite is observed for TANGO2 deficient cells, especially under starvation condition. Also, another thing that we can observe is, for example, the fact that cardiolipin was decreased in TANGO2 deficient cells, but there is some sort of mechanism that can recuperate it in under starvation condition in TANGO2 deficient cells. This is even clearer from this different like way of visualizing it. So we see the lipid amount for lysophosphatidic acid, phosphatidic acid, and cardiolipin under all conditions. And you can see that, as we saw, lysophosphatidic acid increases in TANGO2 depleted cells, while phosphatidic acid decreases when there's no starvation, and cardiolipin too. And while cardiolipin is kind of recuperated at basal level in TANGO2 depleted starved cells. And this might be because there's some rewire metabolism that can involve phosphoglycerol, isophosphoglycerol to produce it, so in starvation conditions. So we saw, in this case, the authors could see that TANGO2 protein is localized in the mitochondria, close to the endoplasmic reticulum, and close to lipid droplets. And when TANGO2 was depleted, the cells were higher and larger lipid droplets. And the larger volume is probably due to this changed lipid metabolism, as we saw, and because we see that TANGO2 depletion is changing the lipid profiles, especially in response to starvation. The study was published in 2023 on eLife and was titled Defects in Lipid Homeostasis Reflect the Function of TANGO2 in Phospholipid and Neutral Lipid Metabolism. The next case study is about lipid droplets and kidney disease in diabetes. This was done by the Heidelberg University Hospital. We saw a lot of participants from there today. And this is one of Europe's largest and most modern medical centers. And in this case, they were trying to understand how diabetes affect lipid droplets in kidney disease and diabetes. We know that kidneys are paired organs, and the functional unit is the nephron. And how does the kidney work? The nephron has two steps of its work. So we have the glomerulus that we see here at the center of this figure that filters the blood, produces this ultrafiltrate, and then the proximal tubule is in this, especially in the S1 segment, will give back all the essential nutrients into the system, so like glucose, amino acids, and proteins, and then will filter the waste that will eventually get out as urine. This system needs energy, and this energy is provided by fatty acids that are stored in the segments 2 and 3 that we see here as lipid droplets. So the authors of the studies were trying to understand if a specific high-fat diet was impacting the kidney function, because we know that when there's a malfunction of the kidney, we have an accumulation of lipid that can cause lipotoxicity and eventually cause even the need for lipid transplant or kidney transplant or dialysis. So in this case, they were inducing diabetes in mice by a low dose of an injection of a low dose of streptozotocin that was destroying the beta pancreatic islet, causing hyperglycemia and thus diabetes. So then the mice were giving a high-fat diet or a controlled diet. The high-fat diet could be of two types. One was rich in monosaturated fatty acid, so it was high in olive oil, so oleic acid, and the other one was high in saturated fatty acids. So that was butter, so it was palmitic acid. We could see here from the graph on the top left, there was an increase of plasma triglyceride after the two dietary interventions. And then, but the most interesting part was that the author wanted to study is how are lipids stored after these high-fat diets. And you can see from the bottom, from part C of this figure, from the section of the kidney, there was a higher accumulation of lipid, especially in the monounsaturated fatty acid diet. This is done by oil. You can see it from oil redox staining. And you can see it better even from the section of the cortex. There, zoom in figure B. And this was also evident. It's also evident from the percent of the stain area that you can see in part D of the graph. So then they thought, is this lipid accumulation also generating damage? So they measured the fibrotic tissue, the percent of fibrotic tissue, in this case with precursorius red staining. And you can see that this was specifically higher in the case of the saturated fatty acid diet. This is even clearer when we normalize this percent of fibrotic tissue by the amount of lipids that were stored. So the oil redox staining that we saw from the figure before. So saturated fatty acids are generating higher damage in the kidneys. So the next step, they translated the study into in vitro. So they wanted to understand if these two treatments, so palmitic acid or oleic acid, or the combination of the two, was causing any it was affecting cell viability. So they measured cytotoxicity. And the first step was doing it with a control. So they measured with BSA, and you could see there was no effect from that. While there was a high effect from the exposure to palmitic acid after 24 hours. So the cells were starting to die, especially after between 4 to 12 hours. And this was a higher response in the later stages, especially at a higher concentration of palmitic acid. While oleic acid was not causing any damage to the cell, so any cell death or any lowering cell viability. And this was the most interesting part, was that if you were adding oleic acid to palmitic acid, oleic acid was able to have a protective effect against the damage of palmitic acid. And the same thing was seen when they counted the red objects, so basically raw species, so reactive oxygen species in the cell culture. And again, you can see that palmitic acid at a concentration dependent response in this. While the use of oleic acid again was able to recuperate some of this negative effect. The next step again was trying to see, since they saw there was an unfolding protein response, so there was some endoplasmic reticulum stress. So they thought there should be also a difference in the lipid metabolism generated by this exposure to palmitic acid or oleic acid. So then they measured the lipid contents of the cell, again with our untargeted approach, and you can see that for example, the levels of free fatty acids were just almost the same. They were kind of lower in the oleic acid treatment. And while, for example, phosphatidic acid was higher when palmitic acid was the treatment, you can see the saturated fatty acids were the higher. And also for diacylglycerides, these were higher for palmitic acid treatment. Well, this is not seen in the graph here, but with oleic acid, we had a higher formation of, for example, triglycerides. So this could be translated to the fact that it was a higher formation of lipid droplets because we have triglycerides, while for palmitic acid, there was more of a response that was generating the precursor of triglyceride, but not triglyceride themselves. So then the authors, in fact, could see that the lipid droplet number was increased with oleic acid treatment and together with the lipid droplet volume. And this was in relation to the higher amount of triglycerides, for example. So to sum up the study, we saw that saturated and unsaturated fatty acids could impact the kidneys differently. So the unsaturated lipids were increased like in storage in the tubule cells, while unsaturated fatty acids were also producing less damage, as we saw from the percent of fibrotic tissue. While saturated fatty acids, so palmitic acid in this case, was producing more damage, as we saw, more fibrotic tissue, more cytotoxicity, and also more endoplasmic reticulum stress. So this study was published in 2022, was titled Reducing Lipid Bilayer Stress by Monounsaturated Fatty Acids Protects Renal Proximal Tubules in Diabetes, was published on eLife. And so this is an important study to understand the impact of diet in diseases like diabetes and the importance of discriminating between different types of fats. The next case study is about lipid metabolism in lipid droplets and how it impacts remyelination. This was done by the Technical University of Munich. And the research question, as we said, is how is lipid droplet metabolism involved in remyelination? So we know that the central nervous system is comprised of neurons, and then there's glial cells like oligodendrocytes, astrocytes, and microglia. We know that oligodendrocytes generate myelin, which covers these axons in the neurons to protect them. And when there's any damage to the myelin sheath, there needs to be remyelination. So this remyelination process is often failing in diseases like Alzheimer's or multiple sclerosis, especially in the later stages of the disease. And some, as we said, like glial cells include also microglia, which are the immune cells of the CNS, and they are fundamental for this remyelination process. So we know that when any myelin damage is present, there's the formation of this myelin debris, which consists of cell fragments, membrane fragments, and certain lipids. And so we see there's also cholesterol in this case, and the higher exposure to cholesterol, for example, to the endoplasmic reticulum will cause endoplasmic reticulum stress. But this is also key for the microglia to sense this cholesterol so they can start this remyelination process. So the author of the study were trying to understand the role that TREM2, which is the triggering receptor expressed on myeloid cells 2, the role that it has in the metabolic pathway involved required for remyelination. So they had postnatal that we see here, or also adult microglia, which were exposed to this myelin debris. And you can see that in the wild type cells, we have an increase in lipid droplets number after 8 hours and 24 hours of exposure, and the same was for adult microglia. But in TREM2 knockout cells, we don't see the same response. So then they wanted to understand again if the lipid metabolism was involved and how this was involved. And you can see here from the graphs on the left side on graph A that in wild type cells, in TREM2 knockout cell, baseline you have a lower amount of cholesterol esters, but this will become eventually cholesterol in the TREM2 knockout cells. And while the opposite is seen in wild type cells. In wild type cells, we have an increase in cholesterol esters. And this is because there's not this response. So the cholesterol is not transformed into cholesterol esters, so it cannot be included into lipid droplets. And the other thing that they observed is an increase in endoplasmic reticulum stress. And this was, they could confirm this by Western blots. So the graph that we see in section B and C. We have an increase of, for example, of the phosphorylated version of eIF2 alpha, which basically means that it's inactivated, so there's less protein translation. And the same is seen with, there's a higher amount of phosphorylated adrenaline terminal kinase, which basically inactivates this phosphorylated eIF2 alpha. So this means that there's a higher endoplasmic reticulum stress because there's a higher exposure to this free cholesterol that is in the system, which is not incorporated into the lipid droplets. So what we see, as I said, there's this myelin debris, and we have the TREM2 wild type microglia, which have the TREM2 receptor and which will sense this cholesterol and can incorporate it into the lipid droplets. While in the knockout cells, this is not possible because the receptor is missing, and this will cause endoplasmic reticulum stress. So to sum up the study, we saw that the process of remyelination will require the clearance of this myelin debris. And this is the fundamental part here, is that the phagocytic function of the microglia relies on the formation of these lipid droplets. Also in this process of sensing the cholesterol and then transformation, esterification of the cholesterol into cholesterol esters. And for this, TREM2 receptor is essential for this process. This study was published on the Journal of Experimental Medicine in 2021 and was titled TREM2-dependent lipid droplet biogenesis in phagocytes is required for remyelination. The last case study that we'll see today is about the role of lipid droplets in ischemic stroke, and this was done by University Medical Center in Göttingen. And the research question, again, as we say, is what is the role of lipid droplets in the resolution of ischemic stroke? We know that stroke is one of the leading cause of disability and death in the world, and is basically when there's deficit to the brain because there's interruption of blood supply to the brain. 85% of all strokes are ischemic stroke, and this is when blood clot or plaque obstructs the cerebral artery, so there's no nutrients, there's no oxygen to the brain. This will cause cell death, injury to the brain, and inflammatory response. And so there's this inflammation response is key to determining the effect and the final extent of the neuronal damage. So as we saw, when there's any damage to the neuron and the axons, we see this myelin debris forming. And in this case, the researcher observed that there's some involvement of this phenotype of microglia, which is called lipid droplet-rich microglia, because they have a higher content and this larger size of lipid droplets, which are basically cause this pro-inflammatory state. And so the authors were trying to investigate more about this microglia phenotype and how it's involved in the resolution of an ischemic stroke. So what they could see is that in hypoxia conditions, there was an increase of this lipid droplet-rich microglia, and this is more specifically seen when you use reoxygenation to the cells, and you can see that there was a higher response, especially peaking at 24 hours and then reaching kind of a plateau there. So there's an increase in this phenotype of microglia. So then they try to understand if what the response is more locally. So they use an in vivo model, and in this case, they use a rodent model with the middle cerebral artery occlusion. And in this case, as you can see from the figure, the lesion area of the brain of this mice was where we have an accumulation of this lipid droplet-rich microglia. And the interesting thing was also that the response was also higher between 3 and 7 days after a stroke. So there's a temporal and spatial response to this. The other thing that they could measure, for example, was an increase in markers of inflammation in this area. So for example, TNF-alpha, interleukin-1 beta, interleukin-6, and a decrease in anti-inflammatory markers like interleukin-10. So then they did, again, in this case, we analyzed the brain tissue with our targeted lipidomics. And you can see the response of the different area of the brain. And you can see that after 7 days, the damaged region is particularly different from the rest of the brain. And this is even clear from the heat map here on the right. If you focus your view on the part that is very red, that's the 7th day after stroke in the core region that was damaged. And you can see that there's an increase of a lot of lipids, especially phospholipids and lysophospholipid. So the authors of the study saw that, microglia accumulate lipid droplets during the ischemic stroke, but it's important for the microglia to be able to go between this pro-inflammatory state to an anti-inflammatory state, because this lipid droplet-rich microglia is linked to worse disease outcomes. And this is also seen in other conditions like Alzheimer's disease, Parkinson, and also in aging. So it's fundamental to understand how this works. And one thing that the authors of the study saw that it was that when they were inhibiting a particular enzyme involved in lipid metabolism, this was some of these negative effects were recuperated. So this is a key study and this is the first step to understand more of this, and this will help in many different applications. The study was published in 2024 in Advanced Science and was titled Dynamic Brain Lipid Profiles Modulate Microglia Lipid Droplet Accumulation and Inflammation Under Ischemic Conditions in Mice. So today, We talked a little bit about what lipid droplets are. We saw how different structure of lipids affect their incorporation into lipid droplets. And finally, we saw some case studies. We saw 4 case studies about the role of lipid droplets in starvation, in kidney disease and diabetes, remyelination, and ischemic stroke.